Okay, tonight, welcome. This is Angela Master. Tonight we're going to talk about Chapter 24, which is Genetics and Genomics. Genetics is referring to, again, DNA. And remember, DNA is contains your genetic code or genes, and those are the recipes for proteins. Again, you are a big protein. Your hair, your nails, your height, eye color, whether you're obese or not, are all regulated by genes. And DNA is what you pass on to your offspring and along with your partner to get new genetic combinations of offspring. So just a refresher, our instruction manuals are in our cells, in our nucleus, and that's our genome. And again, our genome is written in DNA molecules, and DNA consists of adenine, guanine, cytosine, and thymine. And here it's showing the double-strandedness of the DNA molecule. So this is pretty important. The sequence of DNA that encodes a particular protein is a gene. And that's the, long story short, that's what a gene is. A gene is actually made into a protein. And again, your protein is, you are just the machinery of your cells and of you is protein. Genes can vary in form. If you think about people around you, hair color, eye color varies, things like that. Genetics is the study of inheritance characteristics, so you can determine how genes will be passed on to one another. And the genome is the complete set of genetic instructions. So exome, you may not be familiar with this term, or as familiar. You have about 3.2 billion DNA base pairs in the human genome. Only 2% of that codes for genes. So you have between 30 and 40,000 genes in the human genome. The rest of the DNA, they're still trying to figure out what that does. Might regulate how genes are transcribed or things like that. Environmental influences, how genes are expressed. For example, sunlight causes freckles. So if you have the genes for freckles, but you don't go out in the sunlight, even though you have those genes, you may not get freckles or as many freckles. So it's also kind of the nature versus nurture argument. And what this is designed to show you, this is designed to show you, again, your chromosomes contain your DNA. Your DNA is transcribed and translated into proteins. And one of the potential proteins is a channel for chloride ions. And if it is mutated, it is the cystic fibrosis transmembrane conductance regulator protein. In other words, it doesn't work. It cannot transport chloride ions as well as a normal transmembrane conductance regulator protein. And that can lead to a variety of symptoms. Mucus is the most common, so your airways are clogged. Can damage the liver and impair digestion. Also by blocking enzymes. Diabetes is possible. Hard stools may be possible, can affect reproductive, absence of the ductus deferens. Also, your skin is salty, common. It's pretty common, meaning that because you can't transport chloride ions, you're excreting them through your skin. So salty. So having one gene that could potentially be defective or mutated, we'll get to that word in a minute, can lead to a whole myriad of symptoms. and can affect multiple organ systems. So knowing this, genetics has the power to predict. If you are having a baby or have had a baby recently, you'll know you'll talk to a genetic counselor. And again, knowing how genes are distributed in meiosis and the combinations at which they join in fertilization, you can predict your offspring. And that's what genetic counselors do. They predict potential diseases that may occur in your offspring. And they can do this because there are certain modes of inheritance, and these are patterns in which genes are transmitted. So let's talk about some of these patterns. But first, we're going to talk about your genes. And let me get my cursor. Here we are. Remember, you have 23 pairs of 46 chromosomes in humans. That is your diploid number. The first 22 pairs are autosomes. In other words, they determine everything but your gender. And pair 23, pair 23 are your sex chromosomes. Not only does it infer gender, there are other genes on there. And notice, for every trait, remember you get 2 sets of genes, one from mom, one from dad. Okay, so genes can be hundreds of nucleotides, and there are variants, and that's the allele. So the allele is a variation of the gene. You can think about blood types. Some people have type A because they have the gene for A. Some people have type B because they have the gene for B. They're the same protein, but a variation, so an allele. You are homozygous if you have 2 identical alleles. Remember, for each gene, you could also use the word trait here, you get 2 genes. So if they are the same, they're homozygous. If they are different, they are heterozygous. Genotype refers to the genes you get, so that refers to your DNA, whereas phenotype is referring to the traits we see. So you see the phenotypes. And here we have wild-type alleles produce mutations. A mutation is a change in DNA. So wild-type alleles would be quote-unquote normal. So the normal chloride ion channel. And the mutation would be the cystic fibrosis ion channel. And this is kind of referring to Mendelian genetics. Some alleles are dominant. In other words, they can cover up a recessive, and some are recessive. Usually dominant is the capital letter, and recessive is the smaller letter. Recessives can only be expressed if homozygous. So for a recessive trait to be expressed, you need 2 recessive alleles. A dominant trait can be expressed if you have 2 alleles or even just 1 allele. Autosomal means they're carried in the first 22 sets of chromosomes, whereas sex-linked are carried in pair 23. X-linked are carried on the X chromosome. Color blindness would be one. And Y-linked are conditions carried on the Y chromosome. So X-linked, if you think about hemophilia or color blindness, can be passed from mother to son, whereas Y-linked are only passed from father to son. And again, our friend Mendel, genetics, we're talking about recessive inheritance. And one of the traits that happens to be recessive is cystic fibrosis. So the plus, I'm down here, is the normal, and the CF is the allele for the cystic fibrosis gene. Both parents happen to be heterozygous, And if you do genetic counseling and predict the offspring, 21 would be an unaffected non-carrier. 2 would be unaffected but carriers as well, and 1 would actually have the disease. Okay, so every time 2 carrier individuals have a child, they have a 25% chance of having a child with cystic fibrosis and a 75% chance that the child will not have cystic fibrosis. This doesn't mean that if they have 4 children, 3 will be normal and 1 will not. It doesn't mean that at all. It just means every time they have a child, they have a 25% chance of producing a child that has cystic fibrosis. Now, dominant inherits, for you to be dominant, or if it's dominant, that means there's only 1 copy needed. So an example of that would be Huntington's disease, which is a pretty serious disorder. It does cause dementia. However, its symptoms don't come into effect until the late 40s when the person has already reproduced. So you could have this gene and not know it. So again, it's dominant, so only 1 copy is needed. So if you have the mother who is unaffected, so she has 2 normal alleles, and the father who is affected has 1 normal allele and 1 Huntington's dementia or Huntington's disease allele. Every time they have a child, that child has a 50-50 chance of getting Huntington's disease. Now, not every gene follows the Mendelian pattern. There's things that called incomplete dominance and codominance. An example of incomplete dominance is hypercholesteremia. Basically, that means you get, if you are heterozygous for this particular disorder, that means you get an intermediate type. So, for example, people without this causes tumors to grow. People without any of the alleles for that, okay, don't really have really low plasma levels. If you have both, you have really high plasma levels and can potentially get tumors. Whereas if you're heterozygous, you kind of have a phenotype in the middle. Codominance is when you're heterozygous and you have both traits, you have both phenotypes. An example of that would be somebody who is AB blood type. So they have an equal, the A and B alleles are codominant sharing of the phenotype. Notice that A and B are dominant to O. So O, for the majority of people to have the O blood type, you must have the genotype i, meaning that the O is recessive. Now, this is good to keep in mind because even though O is recessive, that does not mean it's the least common. Type O is the most common blood in the United States. Recessive just means it can be masked by another gene. So it has nothing to do with number in the population or anything like that. Other factors that express expression of single genes. This is penetrance, expressivity, and pleiotropy. We'll go to the next slide. Penetrance means that everyone who inherits the disease causing allele has some symptoms. So every time you have the allele, you have some symptoms. Incomplete is that some people do not express the phenotype even though they inherit the alleles. And this an example of this is polydactyly. So polydactyly, or extra digits, has a variable expression. In other words, you can get 2 or 3 digits or a whole digit and just a nub. So here is a child with polydactyl thumbs, and she has almost 2 thumbs. Pleiotropy is when one single genetic disorder has several symptoms. Marfan syndrome is an example. Marfan syndrome occurs, is a mutation in the elastic fibers. The elastic fibers type of connective tissue. And it can produce a variety of symptoms. So even though only one gene is mutated, there's many various symptoms. So many phenotypes are affected. Genetic heterogeneity. That just is referring to a phenotype, and that it could have many, it could be a result of many different types of genes. Deafness is an example. So you can be deaf for many different reasons genetically, not just one. The majority of traits are multifactorial traits. So they're molded by 1 or 2 genes or more, plus environmental factors. Obesity, there's about 85 genes that regulate obesity. Height and skin color are also an example. And other diseases. Heart disease, diabetes, hypertension, cancers are also multifactorial. Okay, again, multifactorial traits are also polygenic traits, meaning they're determined by more than one gene. Okay, as for the chromosome pair 23, or the sex chromosomes, a female has 2 X chromosomes, whereas a male has an X and Y. So again, all eggs or oocytes carry the X chromosome because that's all the mother can give. So that's the only allele she can give. Whereas sperm can either have the X or Y. So the male determines the gender of the child. And why the Y gene determines maleness has to do with a gene called SRY. And this, basically, SRY makes the infant, or the boy, respond to the male hormone testosterone. So the SRY gene with testosterone starts the development of the male characteristics. Now, let's talk about the differences between the X and Y. On the previous slide, you show that the X was a much larger gene, X, or much larger piece of DNA, and it has over 1,500 genes. Whereas on the Y chromosome, there are only 231 genes. So most genes on the X chromosome do not have corresponding alleles on the Y. So this is how some characteristics are passed from mother to son. And then of the 230 chromosomes on the Y, or 230 genes on the Y chromosome, excuse me, some genes are unique to the Y chromosome. Hemophilia is an example of a disorder passed from mother to son. If a mother is a carrier, each time she has a son, there's a 50% chance that she will pass the recessive allele on to him. And again, this allele is only on the X chromosome. So the Y cannot mask the recessive allele. Now, women can also have hemophilia, but they would have to get the recessive allele from both the mother and the father. One recessive allele makes the daughter a carrier. Color blindness is also passed the same way, or follows the same pattern of inheritance. Some traits are sex-linked. In other words, they affect structures that are only found in male or females, beards and the growth of breast, for examples. Some are sex-influenced. An allele can be dominant in one sex and recessive in another, and that's baldness. Heterozygous males are bald, but heterozygous females are not. Chromosome disorders. Chromosome disorders are having extra or less than 46 chromosomes, so having 47 or 45. Euploid is a normal chromosome disorder. However, you can get where you have where you have two sets of chromosomes, and that's pretty devastating. Aha, here's what I want. That's called polyploidy, meaning a drastic increase in number. You have an entire set of extra chromosomes, or entire extra set of chromosomes. Again, most embryos or fetuses die fairly quickly with that abnormality. So nature doesn't really like chromosomal, doesn't like when the number of chromosomes is added to or deleted. Aneuploidy is having a missing chromosome or extra set of chromosomes, and this results from non-disjunction. Non-disjunction or disjunction is when the chromosomal pairs do not separate during meiosis. It can either be the homologous pairs, or it can occur during either the first or second meiotic division. So you can have an offspring with either 47 or 45 chromosomes. 47 is considered trisomy because usually one of the chromosomes you have an extra copy. Monosomy is missing an extra chromosome. Now there's only there's 4 types of trisomy, but in the somatic cells there are 3 types of trisomy, which the infant will survive till birth. 13 and 18, the infants do not live very long. However, trisomy 21 is also known as Down syndrome, and individuals suffering from Down syndrome can live a pretty long life. You can also have trisomy or more in the chromosome pair 23. Turner syndrome, this is the only example of monosomy that I'm aware of, and instead you only get one X. You don't so they're usually raised as females here, although they have lots of shorter in stature in the webbed neck. You can get 2 X chromosomes and a Y. That's Klinefelter syndrome. This is why female athletes sometimes have to undergo genetics testing to make sure they don't have this syndrome. You can be polyfemale, where you can get an extra X or 2 sets of X's. Again, there could be some learning disabilities associated with 1 X, or 3 X's, but the 4 X's have severe mental disabilities. Also, you can receive 2 Ys. They can be tall and have acne, and again, some speech and reading problems, and that's called Jacob's syndrome. So how do you test for chromosomal disorders? There's quite a few ways. The most common is amniocentesis, where they get a section of amniotic fluid, use a big needle to go into the amniotic fluid. This one could lead to miscarriage, so you have to be careful with that one. Chorionic villi sampling is when they remove part of the chorion. This one is less invasive than amniocentesis. Maternal serum markers and fetal cell sorting, they get these from the maternal blood, so it doesn't really harm the fetus at all. And then ultrasound, of course, using sound waves, and that can happen at any time. Okay, and I believe that concludes our lecture here. Thank you, and if there are any questions, please ask.